Thank you to HUD for the invitation to speak here. It's really an honor and a pleasure to be here today. So today I am going to talk to you about congenital disorders of glycosylation. We've heard a lot about that today. Specifically, I'm going to talk to you about N-glycosylation and show you how heterogeneous group of disorders these are, and hopefully introduce you to the zebrafish, as that's the organism that we use to study the set of disorders. And finally end with how studying a rare disease leads to new insights into essential processes, and what my focus is on a new metabolic intersection of glycosylation and P53. This is the cumulative basically number of the glycosylation disorders that have been identified, and in 2013 alone, a new glycosylation disorder was reported every 17 days. You know, I am a pediatric hepatologist by training, and during my fellowship and residency right around here, really CDGs were number 20 out of 20 on the differential diagnosis for babies born with jaundice. It was something that was, you know, we learned about, but was never really anything that we would actually test for. Now in our hepatology clinic, every patient that we have with jaundice, and especially if there's some unknown etiology of neurologic disease as well, all get tested for transferrin isoelectric focusing. So really, the CDGs have been on the forefront of our minds as enterologists, also on our metabolic side, and our geneticists as well. This is a very detailed slide and picture, but what I want to have you take home is that the set of disorders, CDG type 1s, are very diverse in their presentation. And if you just take a snapshot of even this section, you can see that, you know, some patients will present with skeletal defects, some are not reported to have it, the majority will have some in certain different types, but really the clinical presentation runs the gamut of how these patients will be affected with certain symptoms. We initially got into CDG for the liver dysfunction, and HUD had alluded to the MPI CDG patients that present with fibrosis. And so just to give you a brief abridged view of N-glycosylation, you know, we'll start with glucose, and really it's to modify a sugar to make an oligosaccharide. This then gets attached to a lipid-linked anchor called dolichol. It gets flipped into the ER lumen, and then forms a lipid-linked oligosaccharide, which is the major precursor of N-glycosylation, which I'll refer to later on, and then goes through further steps where it gets attached to the asparaginase residue of a protein and goes on to the Golgi for further processing. Now, what I would like to bring your attention to is really the comparison between MPI CDG, which is the patient Max that HUD just presented, and really the reaction, next step, enzymatic reaction next to MPI is PMM2. Now, the juxtaposition of these two diseases are really interesting to us. You know, PMM2 is probably the common CDG. It presents with neurologic defects, seizures, cardiomyopathy, skeletal defects. And really just, you know, upstream of this is the MPI CDG, which is really known as the gastrointestinal type. It's unique in that fact that it's the only CDG that has no neurologic defects. So this contrast was really interesting to us. You know, why does that happen? And we, you know, this is MPI, although we've seen that Mannose therapies may be promising, has been the only one that's proven to really show that it has a treatment. And just as HUD has shown that we can treat with Mannose very gingerly in our concentrations, but and to bypass this MPI defect and proceed on with glycosylation. We screened for these CDGs with transferrin, and this is the maybe of Max, no, of the transferrin SCS page here that shows the change transferrin. Similar to the PMCs are the same, yet the presentations are very different. And we mark the success of our treatments also sometimes by the improvement in transferrin glycosylation. So to even take this to a more simpler route, and we alluded to this in the first session, is, why don't these two present the same? If we presume that it's this defective, glycoprotein that leads to disease, why don't these two diseases have the same exact clinical presentation? So do we think that maybe another possibility could be that it's a specific glycoprotein that's defective that then leads to disease? Or maybe it's a subset of glycoproteins that function, and that function is abnormal, and that leads to some cellular response, and that causes a disease. Alternatively, it could be that if you take the case of MPI-CDG, maybe you have a deficiency of the product or a buildup of the substrate that is lethal or toxic to the organism. So the main questions that we ask is, how does impaired glycosylation in the context of CDG cause disease? Why do CDGs present differently? And most importantly, can we correct this metabolic defect? This brings us to our aim of our project, is really to understand and identify the molecular mechanism underlying CDG. As Hud had mentioned, CDG patients are hypomorphic mutations, and this brings us to why we use the zebrafish, because we have tunable gene knockdown strategies to kind of get a not complete knockout, but a knockdown of these enzymes, albeit not the same functionally as the mutation. Hud's presentation before showed that the mice models have yet to really recapitulate the disease. The knockout was lethal, the knock-ins did not show a phenotype. So this brought us to the zebrafish, and actually this project started as a challenge grant for the stimulus package to develop zebrafish as a model for CDG. And one of the benefits of zebrafish is the ability to do affordable large screen genetic and drug screening. So to tell you a little bit about the zebrafish, it's a transparent vertebrate. They have rapid and external development. You can see in this video, I don't have the mouse here. How do I? Okay. In this video, it's about a 20-second video that will encapsulate 17 hours of development of the zebrafish. And really, the first few hours of zebrafish development is just rapid cellular proliferation. Cells are just trying to divide, and they do that probably every 15 minutes per stage. And so here you'll start to see the tail bud forming. Here you'll see the eye. This is the brain formation. And by 17 hours, you already have a body axis of the zebrafish. You can get over 200 embryos per mating, so the power of statistics for your studies are really great, and it's relatively inexpensive if you compare it to rodent studies. And this is the day 5 zebrafish, where this is my organ of interest, but they have a heart, ear, their gut is here, their swim bladder, and we're able to, do great genetics with zebrafish and humanize our zebrafish. This is a transgenic zebrafish expressing human transferrin, so that we can use, clinical correlates from patients and try and see if those apply to our disease models. So, why did we pick MPI-CDG? So MPI-CDG, I told you, is the gastrointestinal type, and when you treat these with mannose, the symptoms really improve within weeks, except the liver fibrosis continues to progress. And just last year, there was the first paper out to show that there was a successful liver transplant for this patient. And so really, we also wonder, why does this liver disease progress? So there's things that we understand about MPI-CDG, but a lot of things that we don't as well. So we said, okay, well, let's just take a stepwise approach and look at glycosylation, look at cellular responses in the organism. But first, we want to create the zebrafish model. So we use morpholino technology, which is an ATG blocking oligonucleotide. And, MPI activity, we're able to measure just like they do in the patients. And the patients have reported 5 to 18% residual activity. So we have titrated down our morpholino to get us within that range. And our average is about 12%. And this is a picture of a normal zebrafish. Here's the liver, here's the jaw, here's the brain, the eye, and this is where the heart sits, right in front of the liver. And you can see that when we inject our morpholino, we get a multisystemic phenotype in where the liver size is smaller, there's some pericardial edema, there's jaw defects, brain defects, I said this was the gastrointestinal type, but we always get asked the question of why do we see multi-systemic defects? But we think that there's a possibility that there is some sort of maternal mannose in their diet in utero for humans that may be, carrying them through major developmental milestones in utero. And then when they're born, they're, without mannose. But here in the zebrafish, there's no in utero. It's all external development. So perhaps we're seeing these defects that were otherwise partially treated in utero in humans. So really this, we can develop this model, but the thing that this model relies on is, can we rescue these zebrafish with mannose? So zebrafish are really easy to work with, and you either just have control fish water. We treated them with 50 millimolar mannose and glucose as a control. Right, when we inject them with the morpholino, and we look at 4 days, and lo and behold, we see that with mannose treatment, there is significant rescue of the liver size, the body phenotype, and when you look at this, you see significant rescue of the phenotype on scoring with no rescue from the glucose. So this was really great to us. We looked at glycosylation markers, and we found that the LLOs were decreased as well as the MAN6P. So if you block this, the MAN6P was decreased in our morphine, and the LLOs, which was that major precursor, was also decreased. But when you treat with mannose, you get full restoration of LLOs and partial restoration of the MAN6P. We don't really know how long we treat these patients for mannose. HUD shows some great data to say that you need to treat for a long duration, but a lot of these patients, they feel so good so shortly after that they'll stop the mannose. And, then they'll maybe have more diarrhea or their coagulopathy will come back, and then they need to go back on the mannose. So with zebrafish, what we tried is to kind of create a time course and see what window you needed to treat with the mannose. And so we treated all of these fish at day 0. And we found that even if you withdrew the mannose at certain days of development, that you would still get significant rescue with the mannose. So it didn't really matter, but you really needed it to start it right at time 0. We tried the alternative, and we started at different days, but kept it on all the way till day 4. And we found that you actually had to start it at 0, and you could not start it later than time 0, which offered us a little window in saying that this is maybe a crucial time where the mannose needs to be started. What we did notice about these zebrafish were in their phenotype, I showed you the smaller liver, this malformed jaw. This looked like there was a lot of cell death in these phenotypes. So we checked and we found that, programmed cell death or apoptosis, as marked by cleaved caspase 3 staining, was up in these morphins, and that this apoptosis we were able to rescue with the mannose so that it was specifically causing this phenotype. And we quantified this and showed that it was significant. So to summarize this first part, really similar to the CDG patients, our zebrafish deficient in NPI can be phenotypically and biochemically rescued with mannose supplementation. We found that mannose was effective earliest in development, pointing to a critical window of opportunity. And importantly, it provided us a proof of principle to show that you can use zebrafish to study CDGs. So now really our question was great, we have this model, but our question was, what is the underlying pathophysiology? So this is where, we want to look at glycosylation, we want to look at the cellular responses, really take a broad look, do RNA-seq, look at ER stress specifically as that's been associated with PMM2 models, and go on. So surprisingly, we actually used, this is a transgenic which expresses a glycosylated protein in the liver. And when we looked at our MPI morphins, the glycosylation in these liver tissues, which we don't get very much tissue from, but did not look like there was any shift to show hypoglycosylation. And we used tunicamycin, which was presented earlier, and disrupts early glycosylation and is an ER stress inducer. But you see that there's hypoglycosylation here, but really in our morphins, we haven't seen any signs of hypoglycosylation. Some preliminary work by Charlie Gerasi showed that with kinase staining, we also don't see any global major decrease in glycosylation in our morphins, suggesting that maybe this is not critical or really driving our phenotype of our fish. We've looked at ER stress to see whether maybe these abnormally glycosylated proteins aggregate and cause ER stress, and found through RNA-seq that the pathway was mildly, if any, upregulated and did not cause ER stress. So what we did next was really we took a broad survey to say, well, what about when you knock down MPI, what pathways are upregulated? And our top hit was P53. P53, I'll show you a little bit more, but is one of the oldest tumor suppressor genes that have ever been identified. And its related apoptosis pathway markers were also elevated. Glycolysis was down, which I'll hit on next, but really this was surprising to us, that P53 would be involved in MPI knockdown. So P53 really, over the years, it started off by best being known for being a tumor suppressor gene, responding to, DNA replication stress, genotoxic stress, but in the recent years, it's really been shown to be able to respond to mild and physiologic stress, to be a protector and not only an executor here, and has a major metabolic role in glycolysis as well. So we looked at P53 in our zebrafish and found that P53 protein levels were increased and stabilized over development from day 1 to day 4. And we looked at all of its targets here are upregulated as well. And that brings us to kind of a simple model that when you downregulate or you decrease MPI, that it upregulates P53, leads to apoptosis, which I showed you with the cleaved caspase 3. And then this is responsible for our phenotype. Now, we looked at our zebrafish and said, okay, can we rescue our MPI morphins with P53 knockdown? And lo and behold, if you use P53 morpholino to knock that down, or you inject MPI morpholino into the P53 mutant, you see full rescue of the phenotype, as shown here. And what's even more interesting is that when you look at these, compare the morphins to the rescued zebrafish, you find that their MPI activity is still the same. So, here, these rescued fish have still only 7% MPI activity, which really was more evidence showing that glycosylation was not the driver of MPI knockdown in our fish. And to see if this P53 upregulation was specific to MPI or was it generalizable to glycosylation disruption or ER stress, we injected PMM2 morpholino into P53 mutants, and we also treated P53 mutants with tunicamycin and found no rescue at all. So this seems to be that P53 and MPI have a specific interaction. Furthermore, in our RNA-seq data, we showed that not only is P53 upregulated with MPI knockdown, but that glycosylation genes didn't really change. Whether or not this had something to do with salvage and you were able to, create enough MAN6P to not change glycosylation, but really what we found was that glycolysis was decreased. Globally decreased in our RNA-seq data and some preliminary metabolomics data. So why is this important? To put this in a bigger scheme of things, the balance between glycolysis and the fact that MPI now is linked to P53 and glycolysis regulation, glycolysis in embryonic development is so important to just proliferating cells. When you have cells dividing, like I showed you in the video, glycolysis is essential. It's not until you start to differentiate and have organogenesis that there's this embryonic shift in metabolism, where then you shift to more oxidative phosphorylation. This same shift between oxidative and phosphorylation and glycolysis is essential and basically the crux of what we call the Warburg effect in cancer, where normal cells, undergo really oxidative phosphorylation as it produces much more ATP. But when you have cancerous cells, these preferentially now upregulate glycolysis. So our hypothesis that is shared by many is that really in cancer, maybe you're getting this shift back to earlier embryogenesis, where you just want lots of cellular proliferation, and we think that MPI plays an important role in this. So what our model is, and what I've hoped that I've shown you today, is that phosphate isomerase MPI really sits between this, is a fulcrum, important modulator between, not only glycosylation, but also it upregulates P53, and that we see its target genes are upregulated as well. That leads to apoptosis. We don't know that, whether a specific glycoprotein may still be contributing to this, and the abnormal glycosylation of that may lead to this apoptosis. But really, this role of glycolysis and this downregulation of glycolysis seems really important to this model, in that we think that if you have downregulation of glycolysis, you're not able to make the energy that the organism needs. You have ATP depletion, which then leads to the apoptosis. And this downregulation of glycolysis may be mediated specifically by MPI, or we know that there's a lot of evidence that shows that P53 downregulates glycolysis, and that, this arrow here, we have our new evidence that this arrow may be bidirectional, that MPI and P53 may be directly interacting with each other. So in summary, CDGs are very heterogeneous in presentation, but they have an unclear pathophysiology. And we have shown that zebrafish can be used to study CDG with similar clinical cornerstones that may reveal developmental windows that are important for treatment. We see that MPI surprisingly upregulates P53 and its apoptosis target genes, and that MPI CDG may not solely be attributable to glycosylation status, rather may be driven by P53 and decreased glycolysis. So this is, I want to just acknowledge the lab, and we work very closely with Kirsten Sadler-Depley, who also runs our zebrafish facility. Hud Freeze, of course, for his guidance and sources of funding. And I'd be happy to take any questions. No, it's transcriptionally it's not that upregulated, maybe twofold, but it's on the protein level. And we found that, we looked at MPI and downregulation of MPI upregulates P53, but now we've looked at it from the other way. Can P53 upregulation downregulate MPI? And the model that we're using for the zebrafish now is to irradiate them and give them UV, and we upregulate P53, and we find a dose-dependent decrease in MPI activity and protein level 2. So that's where I think we're finding this really exciting interaction between P53 and MPI, and that, P53 has always been this tumor suppressor, now shown in glycolysis, but actually can be involved in sensing mannose metabolism as well. So that's, like I showed you, where embryonic versus, differentiated versus cancer. So the way we're addressing that is looking at primary hepatocytes and liver cancer cells, and looking to see how those pathways are different in development versus cancerous cells. And it may be different. It probably will be.
Well, I have a question. Are you planning on making some of these model?
Yeah, that's a great question. So in terms of to get at whether knocking down with a morpholino, like I mentioned, is not the same as the human mutation. So the problem with human mutations in MPI, as we learned from the mouse model, is not only it's hard. Sometimes even those mutations don't recapitulate in the mice too. And so we have thought about that. We right now have talons running, so we're going to do a mutation that way. And what Charlie's working on now is to see whether this P53 MPI interaction is really essential. It's basically from the catalytic site or the protein as a whole, and is creating mutants in the catalytic site to see if we see the same thing. You have a question?
I assume the MPI is the cytoplasm in the protein.
I'm sorry.
MPI is located in the cytoplasm.
Mm-hmm.
Is P53 primarily located in the nucleus?
P53 is primarily localized in the nucleus, and
So how do you do that?
this is a hard question. So we actually sometimes see that this is preliminary data, and not, we need to repeat it, but there's some data by Charlie shows that MPI may be actually nuclear localized too in certain states. So I don't know, I think it's an ongoing experiments, and we're doing some IPs now and immunofluorescence to look at localization as well.
So
since all of the P53 regulation occurs in the first 24 hours, that's the place where you can rescue the nanos. If all of that is interrelated, then how meaningful is it to measure metabolizing oligo and protein glycosylation at 4 or 5 days? I mean, obviously,
yes.
That's, is it still relevant? How do we look at that?
I think we need to, the problem with, we have the yolk is a big problem. So actually when we were looking at the LLOs and the, the glycosylation, it ended up that we were actually just starting the model. So it was more as we were developing this. But now that we know that this is a big window that we're looking at right at the first 24 hours, we're starting to do more early embryo work and de-yolking these things and trying to see if we can test for those things too. That's right. We don't know.
So glycolysis, aerobic glycolysis is turned on by that pathway being altered?
I didn't see that in the RNA-seq analysis. We looked at that a lot, and it didn't pan out right now, but we're trying to look at more of the metabolomics that we're working with Mayo to do that, and maybe to give us some sort of sense of, but that's also steady state. So really flux studies would be really key to seeing which pathways are really changed acutely. We also think that there's a difference between acute MPI loss versus chronic MPI loss. So, we're working with HUD and Charlie's doing some of the things looking at patient fibroblasts to see, if they're notorious for not growing and proliferating very easily, maybe we knock down P53 and then can we restore that phenotype as well. But that's also a chronic loss of MPI, which may be much different than the morpholino knockdown too.
So another quick follow-up to that. So the other thing is if you're thinking that you're altering fructose 6-phosphate, fructose 6-phosphate is also key to go into. I
know, so that's what I'm learning today too. And P53 is not N-glycosylated, but it is predicted to have O-glycosylation. So yes, I was really interested to see fructose 6-phosphate popping up many times today, and because I think that that's really a key in terms of what the cell is sensing, levels of fructose 6-phosphate.